Welcome to Relaxing Sleepy Science. You've arrived at a place where deep research meets a quiet mind. Thank you for listening to this compilation of Relaxing Sleepy Science. Tonight we explore the intricate history of gene expression. We will move through the human genome to understand how the process of proteins shaped life as we know it. If you enjoy these gentle journeys, I invite you to like, subscribe, or share a thought below. But for now, there is nothing you need to do but relax. Soften. In the calm of a lab, a single cell lies in wait. It's a human cell, tiny yet vast in its potential. It holds within it the blueprint of life, the human genome, wrapped in a neat double helix. This quiet sentinel contains approximately 20,000 to 25,000 protein-coding genes, each a promise of life's intricacies. Its stillness is a world of biological processes ready to unfold. The cell, a microcosm of life, is not silent. It hums with a stillness, a rhythm that has echoed through generations. The promise of these genes lies dormant, waiting for the right signal to awaken. Its solitude is a testament to life's complexities, a collection of chemical reactions and biological processes. As we zoom in, the cell's nucleus comes into focus. It's here that the genome resides, a library of life's instructions. The DNA, a delicate coil of information, begins to unravel. It's a slow process of transcription. The DNA, in its quiet elegance, is a master of disguise, hiding in plain sight. The cell's nucleus, a quiet sanctuary, is not a solitary entity. It's a bustling hub of activity, a microcosm of life's processes. The DNA, as it unravels, begins to dictate the process of life. The RNA, a faithful messenger, begins its journey carrying the promise of the gene. Its stillness is a testament to life's relentless pursuit of existence. As the scene slowly changes away from the cell, we carry with us the promise of gene expression. Its quiet solitude is a symbol of life's potential. It's a promise held within the coils of DNA, waiting to be unwound. Its stillness is a quiet revolution, a silent evolution. And so, our journey into the promise of gene expression begins. As the transcription unfolds, the DNA, in its quiet elegance, begins to unravel. It's a slow process, a rhythmic uncoiling of the delicate coil, like the unrolling of a parchment filled with life's intricate instructions. The DNA, a master of disguise, hides in plain sight, its structure a collection of chemical bonds and nucleotide bases. You notice the hydrogen bonds like tiny silent musicians playing their part in stabilizing the double helix. They hum a quiet tune, a song of balance and precision, as they connect the nucleotide bases, the A's to the T's, the C's to the G's. This dance of bonds, a ballet of atoms, is the secret to the DNA's structure, its strength, and its stability. The DNA, as it unravels, reveals its secrets one base pair at a time. It's a quiet process, a slow reveal like the turning of a page in an ancient tome. Each unraveled base pair is a whisper of a promise, a hint of the gene that lies dormant, waiting for the right signal to awaken. The cell's nucleus, a quiet sanctuary, is not a solitary entity. It's a bustling hub of activity, a microcosm of life's processes. The DNA, as it unravels, begins to dictate the dance of transcription, a quiet revolution that echoes through the cell, a silent evolution that has been playing out since the beginning of life. And so, our journey into the promise of gene expression continues, one unraveled base pair at a time. In the gentle unraveling of DNA, you find yourself drifting through the quiet sanctuary of the cell's nucleus, a hub of life's processes. The DNA continues to reveal its secrets, dictating the transcription process, a silent revolution that has been playing out since the emergence of life. You notice that transcription factors approaching the stage of the gene. They are the choreographers of this intricate ballet, their role crucial in determining whether the gene expression will commence or remain dormant. The transcription factors are equipped with specific domains that allow them to recognize and bind to particular sequences of DNA, much like a key fitting into a lock. These sequences, known as response elements, are the instructions written into the genome, guiding the transcription factors on where to attach and what role to play. It's a dance of specificity, a collection of recognition. As the transcription factors bind to their respective response elements, they begin to orchestrate the assembly of the transcription machinery. They act as directors, signaling to the various components, the RNA polymerase, the general transcription factors, and the initiation factors, to take their places on the stage. It's a complex ballet, a delicate interplay of proteins and DNA, each with their unique role to ensure the accurate and efficient transcription of the gene. The RNA polymerase begins to roll along the DNA strand, unzipping the double helix and reading the sequence of nucleotides, much like a musician playing a score. As it moves, it converts the language of DNA into the language of RNA, a process called transcription. It's a slow and steady process, a rhythmic uncoiling of the DNA's secrets, one nucleotide at a time. The general transcription factors assist the RNA polymerase, helping it to initiate and elongate the transcription process. The initiation factors prepare the stage for the transcription machinery, ensuring that everything is in place for the dance to begin. This intricate dance of transcription, guided by the transcription factors, is the key to gene expression. It's a dance that has been playing out in every cell, in every organism since the emergence of life. It's a dance that is as complex and beautiful as it is essential to life as we know it. And so, our journey into the promise of gene expression continues, one transcribed nucleotide at a time. In the gentle unraveling of the double helix, the RNA polymerase, our steady conductor, continues its measured march along the DNA strand. It's a slow, deliberate process, a rhythmic uncoiling of life's secrets, one nucleotide at a time. As it moves, it's not alone. The general transcription factors assist the polymerase, guiding it through the intricate ballet of transcription. They play their part, ensuring each step is precise, each note in the collection of gene expression played true. You notice the polymerase slowing, approaching a particular sequence on the DNA strand. Here, the cap, a small yet significant structure, awaits. It's a simple addition, just a small methyl group at the 5' end of the mRNA, but it carries great influence. The cap signals to the translation machinery that this mRNA is ready, that it's time to begin the next phase of gene expression. The cap stands guard over the mRNA, protecting it from degrading enzymes that would seek to shorten its life. It's a subtle role, but a crucial one. Without the cap, the mRNA would be fragile, its message lost before it could be translated. But with the cap in place, the mRNA is steadied, its message preserved until the time is right for translation. As the polymerase continues, you notice the cap growing, expanding like a tiny glowing bubble at the end of the mRNA. It's a quiet, unassuming process, but it's a key moment in the life of the mRNA. With the cap in place, the mRNA is ready, its message secure, its future assured. It's a small step in the delicate dance of gene expression, but it's a critical one, a testament to the intricate beauty of life at its most fundamental level. And so the process continues, one capped mRNA at a time. You notice the polymerase, a molecular ballet dancer, slowing its meticulous waltz across the DNA strand. It approaches a particular sequence, a pause in the collection of transcription. Here, a small yet mighty structure awaits, the cap, a simple methyl group at the 5' end of the mRNA. It's a quiet sentinel, a subtle addition, but it carries great influence. The cap, like a conductor's baton, signals to the translation machinery that this mRNA is ready, that it's time for the next phase of gene expression. As the polymerase continues its dance, you notice the cap growing, expanding like a tiny glowing bubble at the end of the mRNA. It's a quiet, unassuming process, but it's a key moment in the life of the mRNA. With the cap in place, the mRNA is steadied, its message preserved until the time is right for translation. The cap, our quiet guardian, stands guard over the mRNA, protecting it from degrading enzymes that would seek to shorten its life. Without the cap, the mRNA would be fragile, its message lost before it could be translated. But with the cap in place, the mRNA is ready, its message secure, its future assured. You drift closer, drawn by the quiet efficiency of the process. The mRNA, now capped, begins to unravel, its nucleotides stretching out like a string of pearls. It's a slow, deliberate process, each nucleotide unwinding from its neighbor with a quiet, almost imperceptible click. The mRNA, now free from the confines of the DNA, floats away, ready to carry its message to the ribosomes. The ribosomes, tiny factories nestled within the cytoplasm, await their arrival. They are silent, patient, their surfaces marked with intricate patterns of RNA and protein. They are the next phase of the gene expression journey, the interpreters of the mRNA's message. As the mRNA approaches, one of the ribosome's subunits reaches out, wrapping around the mRNA like a welcoming embrace. It's a gentle, careful motion, a testament to the intricate, delicate beauty of life at its most fundamental level. The dance continues, one capped mRNA at a time. You find yourself in a vast, dimly lit chamber, a cathedral of cellular architecture. The air is thick with the hum of activity, a collection of molecular dance. You're drawn to a delicate spiraling staircase, the DNA helix as it ascends towards the nucleus. At its base, the polymerase, a meticulous scribe, poises its quill, ready to transcribe the genetic code into a fledgling mRNA. The polymerase begins its dance, stepping along the rungs of the helix, its quill dipping into the inkwell of nucleotides. It's a slow, deliberate process, each step echoing with a soft, almost imperceptible click. The mRNA, a nascent strand, unravels behind the polymerase, its sequence dictated by the DNA's code. A small, unassuming figure approaches. It's the capping enzyme, a quiet, efficient worker. It carries a tiny cap, a guanosine nucleotide, a guardian for the mRNA. The enzyme leans in, its hands deftly attaching the cap to the 5' end of the mRNA. It's a quiet, unassuming process, but it's a key moment in the life of the mRNA. With the cap in place, the mRNA is steadied, its message preserved until the time is right for translation. The cap stands guard over the mRNA, protecting it from degrading enzymes that would seek to shorten its life. Without the cap, the mRNA would be fragile, its message lost before it could be translated. But with the cap in place, the mRNA is ready, its message secure, its future assured. You drift closer, drawn by the quiet efficiency of the process. The mRNA, now capped, begins to unravel, its nucleotides stretching out like a string of pearls. It's a slow, deliberate process, each nucleotide unwinding from its neighbor with a quiet, almost imperceptible click. The mRNA, now free from the confines of the DNA, floats away, ready to carry its message to the ribosomes. The ribosomes, tiny factories nestled within the cytoplasm, await their arrival. They are silent, patient, their surfaces marked with intricate patterns of RNA and protein. They are the next phase of the gene expression journey, the interpreters of the mRNA's message. As the mRNA approaches, one of the ribosome subunits reaches out, wrapping around the mRNA like a welcoming embrace. It's a gentle, careful motion, a testament to the intricate, delicate beauty of life at its most fundamental level. The dance continues, one capped mRNA at a time. As the mRNA begins to unravel, its nucleotides stretching out like a string of pearls, you notice a quiet transformation. The cap, our guardian, has done its job steadying the mRNA, preserving its message. Now, it's time for another process to take the stage. You drift closer, drawn by the intricate interaction of life at its most fundamental level. You're floating through a dimly lit landscape, the air thick with the hum of activity. This is the realm of the epigenetic regulators, the quiet influencers of gene expression. Here, chemical modifications to DNA and histone proteins play out their silent roles. You approach a cluster of these influencers, their surfaces marked with intricate patterns of methyl and acetyl groups. One of these influencers reaches out, its methyl group extending like a tentative finger. It hovers over a specific region of DNA, a gene that has been silent, its message unheard. The methyl group begins to add its mark, a small chemical modification that will have profound consequences. Methylation, the slow, deliberate process of adding a methyl group to DNA, can silence a gene, muffle its message. But methylation is not the only player in this chemical ballet. Another influencer, its surface marked with acetyl groups, drifts into view. This is acetylation, the process of adding an acetyl group to histone proteins, a process that can loosen the packing of DNA, allowing genes to be heard once more. The acetyl group, our quiet key turner, begins to unlock the DNA, its message now freed to be read. You notice the intricate interplay between these influencers, their actions a delicate, precise dance. Methylation can silence a gene, but acetylation can unlock it once more. These influencers, these chemical modifications, are the quiet conductors of the gene expression group, their actions shaping the collection of life. This dance of influencers has been playing out for billions of years, its steps as old as life itself. It has shaped the course of evolution, guiding the development of every organism. It has played its part in the history of every cell, every organ, every creature. And it continues to play its role today in every cell of your body, in every moment of your life. But this dance is not without its mysteries. While we understand the steps of methylation and acetylation, we do not yet know all the players in this dance. There are other chemical modifications, other influencers, their roles yet to be fully understood. The interaction of life, it seems, is far from over. You drift through a landscape of DNA, its twisted ladder stretching out in all directions. the surface you left behind, marked with acetyl groups, drifts into view. This is acetylation, the quiet process of adding an acetyl group to histone proteins, a process that can loosen the packing of DNA, allowing genes to be heard once more. The acetyl group, your quiet key turner, begins to unlock the DNA, its message now freed to be read. You notice the intricate interplay between these factors, their actions a delicate, precise dance. Methylation can silence a gene, but acetylation can unlock it once more. These factors, these chemical modifications, are the quiet conductors of the gene expression group, their actions shaping the collection of life. But this dance of factors is not the only conductor. There are others, each with their own unique rhythm. Specificity factors, repressors, activators, enhancers, and silencers all play their part, their actions as varied as their names. They guide the transcription process like a chorus of quiet guides. The specificity factors, with their precise, targeted touch, ensure that the right gene is activated at the right time. They are the librarians of the cell, guiding the transcription machinery to the correct shelf. The repressors, with their heavy, silencing hands, push down on the gene, muffling its message. The activators, with their light, encouraging touch, lift the gene up, amplifying its signal. The enhancers, with their far-reaching influence, boost the gene's message from afar. The silencers, with their quiet, persistent presence, dampen the gene's voice. Each of these factors, each of these mechanisms has been playing its part for billions of years. They have shaped the course of evolution, guiding the development of every organism, from the simplest bacterium to the most complex mammal. They have played their part in the history of every cell, every organ, every creature. And they continue to play their role today, in every cell of your body, in every moment of your life. Yet this dance is not without its mysteries. While we understand the steps of methylation and acetylation, the roles of the specificity factors, repressors, activators, enhancers, and silencers, there are other players in this dance. Other chemical modifications, other factors, their roles yet to be fully understood. The interaction of life, it seems, is far from over. There is still much to explore, much to understand in the calm, intricate world of gene regulation. In the gentle sway of the cell, the specificity factors, with their precise, targeted touch, guide the transcription machinery to the correct gene. They are the librarians of the cell, each one knowing its path, its purpose in the complex library of DNA. The repressors, heavy with their silencing influence, press down on the genes, muffling their messages, while the activators, light and encouraging, lift the genes up, amplifying their signals. The enhancers, far-reaching in their influence, boost the gene's messages from afar, and the silencers, quiet and persistent, dampen their voices. You notice the chromatin, the complex of DNA and proteins, unraveling slightly like a slow-motion wave as the transcription machinery approaches. The DNA, a double helix of nucleotides, each a specific sequence of A, T, C, or G, begins to unwind, revealing the genetic code within. The chromatin remodelers, like tiny, delicate architects, reshape the structure of the chromatin, making some genes more accessible, others less so. The RNA polymerase, the chief transcriber, approaches the gene, ready to copy the genetic message from DNA to RNA. It is a complex machine with many parts, each with its specific role. The polymerase itself, a large multi-subunit enzyme, begins to move along the DNA strand, reading the sequence, transcribing it onto a growing chain of RNA nucleotides. As the polymerase moves, you notice the methyltransferases, the quiet, precise chemical engineers, adding methyl groups to specific locations on the DNA. Methylation, a delicate dance of addition and subtraction, can silence genes, dampening their messages or activate them, amplifying their signals. These methyltransferases, with their tiny, targeted tools, are the tiers of the epigenetic landscape, shaping the course of gene expression. The histone acetyltransferases, their cousins in chemical modification, add acetyl groups to the histone proteins, loosening the chromatin's grip, making the DNA more accessible. The histone deacetylases, in turn, remove these acetyl groups, tightening the chromatin's hold, silencing the genes within. The specificity factors, in their delicate dance, bind to the specific DNA sequences known as promoter regions, marking the start of a gene. They guide the transcription machinery, ensuring that the right gene is activated at the right time. They are the conductors of the collection of gene expression, each one playing its part in the intricate, delicate interaction of life. Yet this dance is far from simple, far from fully understood. You notice the introns, the non-coding regions of DNA, as the RNA is transcribed. The introns, once thought to be mere junk, are now known to play crucial roles in gene regulation. They are the silent sentinels of the genome, their functions still largely mysterious, their secrets yet to be fully revealed. The exons, the coding regions of DNA, are joined together, forming a continuous chain of RNA ready to be translated into protein. The RNA, once transcribed, must be processed before it can be translated. The capping machinery, a complex of proteins, adds a cap to the 5' end of the RNA, marking it as mature, ready for translation. The splicing machinery, a complex of proteins and small nuclear RNAs, removes the introns, joining the exons together, forming a mature mRNA. The polytail, a long sequence of adenine nucleotides, is added to the 3' end of the RNA by the polyadenylation machinery. This tail signals to the translation machinery that the RNA is ready to be translated into protein. The mRNA, now mature, ready for translation, begins its journey to the ribosome, the site of protein synthesis. The ribosome, a complex machine, begins to read the sequence of nucleotides on the mRNA, translating it into a sequence of amino acids, the building blocks of protein. The tRNAs, the tiny, delicate carriers of amino acids, bring each amino acid to the ribosome, where it is added to the growing chain of protein. The elongation factors, the quiet, efficient workers, help to move the mRNA through the ribosome, one codon at a time. The release factors signal to the ribosome that the protein has been fully synthesized. The ribosome disassembles, the mRNA is degraded, and the protein, now fully formed, begins its journey to its destination, ready to carry out its specific role in the cell. This dance of gene expression, this intricate, delicate ballet, has been playing out for billions of years, shaping the course of evolution, guiding the development of every organism. It has played its part in the history of every cell, every organ, every creature. And it continues to play its role today in every cell of your body, in every moment of your life. Yet this dance is not without its mysteries. There is still much to explore, much to understand in the calm, intricate world of gene expression. The interaction of life, it seems, is far from over. As the mRNA, the delicate messenger, unwinds from the nucleus, it finds itself in a world of activity. The cytoplasm, a bustling landscape teeming with tiny, intricate workers, is a collection of life. You are this mRNA, drifting through this cellular metropolis, your sequence of nucleotides, the silent instructions for a protein, a whisper in the calm hum of life. You notice the ribosome, a complex machine like a city square where many paths converge. It's a gathering place, a meeting point for the many players in this dance of gene expression. Here, the tRNAs, the tiny, delicate carriers of amino acids, wait patiently, ready to bring each amino acid to the ribosome, where it is added to the growing chain of protein. Silent workers, the quiet hands that build the proteins that shape life. The elongation factors, the quiet guides, help to move the mRNA through the ribosome, one codon at a time. Traffic controllers, the quiet guides, ensuring the smooth flow of information. The release factors, the silent sentinels, stand at the end of the message, signaling to the ribosome that the protein has been fully synthesized. Quiet watchers, the silent guardians of the end of life's instructions. You notice the chemical modifications, the quiet subtle changes to the DNA and the histones. Quiet conversations that shape life. Methylation, acetylation, they are the quiet hands that write the story of life, one gene at a time. Quiet scribes, the silent chroniclers of life's journey. You notice the intricate world of gene regulation, the delicate interaction of life. It is a world of silence and subtlety, a world of quiet conversations and silent agreements. It is a world of stillness, a world of quiet strength. It is a world that shapes life, a world that guides life, a world that is life. This dance of gene expression, this intricate, delicate ballet, has been playing out for billions of years. It has shaped the course of evolution, guided the development of every organism, from the simplest bacterium to the most complex mammal. It has played its part in the history of every cell, every organ, every creature. And it continues to play its role today in every cell of your body, in every moment of your life. Yet, this dance is not without its mysteries. There is still much to explore, much to understand in the calm, intricate world of gene expression. The interaction of life, it seems, is far from over. You're floating, weightless, in an open space. It's not empty, though. It's filled with a quiet hum, a silent song of life. You're drifting to the intricate ballet of gene regulation, the quiet interaction of life's instructions. The stage is set, a vast, sprawling genome, a collection of genes, waiting to be played. The curtains have yet to rise, but the group is tuning, ready to perform the oldest show on Earth. The scene is not static. It's a living tapestry, woven with the threads of time. You notice the regions, the genes, each one a potential actor in this ancient drama. They're not idle, waiting for their cues. They're active, engaged in a constant conversation with their neighbors. This is not a dance of equals, though. Some genes are silent, their parts yet to come. Others are in full swing, their proteins already in the spotlight. You see, the machinery, the molecular players in this dance. Transcription factors, activators, repressors, they're all there, taking their places. They're not human, these dancers. They're not. Even animals. They're molecules, tiny, intricate, each one a master of its craft. They're not dancing for applause. They're dancing for life. time is fluid. Here, it ebbs and flows like the tide. You're in the deep past, billions of years ago, watching the first genes take their places on the stage. But you're also in the present, in every cell of your body, where this dance is happening right now. You're in the future too, in every unborn child, where this dance will continue, generation after generation. This dance is not without its rules. There are conductors here, master regulators, genes that control the expression of other genes. They're not dictators though. They're more like wise elders guiding the dance, ensuring it stays in rhythm. They're not perfect, these conductors. Sometimes they falter. Sometimes they lead the dance astray. When that happens, S can take the stage. Cancer, neurodegenerative disorders, metabolic disorders, they're all part of this dance too. Their genes gone rogue, their movements chaotic, destructive. But even in these performances, there's hope. There are always genes that can step in, take control, bring the dance back into rhythm. That's the beauty of this dance. It's not a one-time show. It's a constant, ever-evolving performance. And it's happening right now, in you, in every cell, in every moment. You're in the gentle sway of cellular machinery, drifting through the intricate ballet of gene regulation. The stage is set, not in the calm corners of every cell, but in the calm corners of every cell, where the interaction of life is choreographed. You notice the genes, not as silent directors, but as quiet directors, each one taking its place, ready to conduct the collection of protein production. They're not conducting for applause, nor are they in a quiet stillness of the grove. They're in a vast, intricate web of interaction, a network that spans from the deep past to the present, from the simplest bacterium to the most complex human. You find yourself in the calm before the dawn of life on Earth. The genes, in their primitive forms, are taking their first tentative steps on the stage of evolution. They're not alone, though. The environment, the other molecules, the cellular structures, they're all part of this dance too. The dance is not just about the genes, but about their interactions, their communications, their regulations. It's a complex pas de deux, a dialogue between genes and their environment. The genes, they're not just passive participants. They're active guides, influencing not just their own expression, but that of others too. They're not dictators though. They're more like wise elders, guiding the dance, ensuring it stays in rhythm. They're not perfect, these guides. Sometimes they falter. Sometimes they lead the dance astray. When that happens, shh can take the stage. Cancer, neurodegenerative disorders, metabolic disorders, they're all part of this dance too. Their genes gone rogue, their movements chaotic, destructive. But even in these dark performances, there's hope. There are always genes that can step in, take control, bring the dance back into rhythm. You're not just a spectator though. You're a participant too. Your body, it's not a silent vessel. It's a living, breathing, ever-changing ecosystem. The dance of gene regulation, it's not happening out there in some distant, unknowable realm. It's happening right here, right now, in you, in every cell, in every moment. It's a constant, ever-evolving performance, and you're not just in the audience. You are on stage. You're part of the dance. This dance of gene regulation, it seems, is far from over. It's been happening for billions of years, and it's a dance that will continue long after you're gone. It's as old as life itself and as new as the present moment. It's not just about the past, not just about the present, but about the future too. It's about you, about me, about us, about all living things. It's about life, about its origins, about its continuance, about its evolution. It's about the very essence of who we are and where we come from and where we're going. It's about the mystery of life and the wonder of existence. And it's happening right now, in you, in every cell, in every moment. In the intricate dance of gene regulation, you find yourself drifting through the labyrinthine landscape of the human genome. That has been evolving with life itself, adapting to changing conditions, optimizing organismal fitness. You notice the chromosomes, like sprawling ancient trees, their branches stretching out, bearing the weight of countless genes, each a standing figure, waiting for its moment to step into the spotlight of expression. The genome, it seems, is not a static entity, but a dynamic, ever-changing ecosystem. The regulatory regions, like quiet conductors, wave their batons, directing the group of transcription factors, activators, and repressors. They decide when a gene steps onto the stage, when it into the shadows. You notice the epigenetic modifications, like intricate tattoos on the surface of the DNA, altering its structure, influencing its expression. Methyl groups like tiny silent cells stand guard over genes, suppressing their expression. Histones, like ancient wise philosophers, wrap around the DNA, their tails bearing the marks of experience, influencing how the dance unfolds. The dance of gene regulation is not just about the past, not just about the present, but about the future too. It's about the silent whispers of evolution, the quiet adaptations that have allowed life to thrive in the face of change. It's about the intricate, delicate balance that allows cells to function in harmony, allowing organs to carry out their duties, allowing life to continue, generation after generation. As you drift through this vast, quiet landscape, you notice the stillness of gene regulation, the way it shapes every aspect of life, from the smallest microbe to the most complex organism. That is happening right now, in you, in every cell, in every moment. That is as old as life itself and as new as the present moment. That is about the essence of who we are and where we come from and where we're going. That is about the mystery of life and the wonder of existence. As you drift through the quiet landscape of a cell, you notice the intricate dance of gene regulation. It's a ballet that's been happening since the dawn of life, a rhythm that's as old as the first single-celled organisms. You've seen the epigenetic modifications, the methyl groups and histone tails like silent choreographers dictating the movements of the genes. But this dance is not just about the present. It's about the future too. Imagine the future of gene regulation, a time when we understand this dance so well, we can join in. Future research is about learning the steps, about becoming a partner in this ancient ballet. It's about developing novel therapeutic strategies, learning to turn genes on and off like a switch. You notice the researchers, future scientists, learning the language of the genes, the alphabet of life. They're deciphering the codes, understanding the whispers of evolution. Read the silent tattoos on the DNA, the intricate patterns of methylation, the histone marks of experience. Predict the dance to anticipate the rhythm. They're integrating genomics and epigenomics, understanding how the past, the present, and the future of gene regulation are all intertwined. See the dance not just as a series of steps, but as a complex, interconnected system. Influence the dance to change the rhythm. They're developing new therapeutic strategies, learning to turn genes on and off with precision. Treat disorders, not by targeting symptoms, but by understanding the root cause, the dance of the genes. This is the future of gene regulation, a time when we're not just observers, but participants. A time when we understand the dance so well, we can join in. A time when we can influence the rhythm, change the steps, and perhaps even choreograph new ones. And it's happening right now, in the calm landscape of a cell, in every moment, in every generation, in you. You're drifting through the cellular landscape, a silent spectator in this ancient ballet. The transcription factors, your guides, approach the DNA, their hands outstretched, fingers dancing with the specific sequences they recognize. They're like keys fitting into locks, unlocking the potential of the genes. You notice the RNA polymerase, a complex machine assembling at the starting point of a gene. It's a meticulous process, like a craftsman carving a sculpture, cutting away the non-coding sequences, the introns, leaving behind the exons, the blueprint for a protein. The polymerase moves along the DNA, reading the code 3 letters at a time, transcribing it onto the RNA strand. The RNA, once freed from the DNA's grasp, undergoes its own transformation. It's capped at one end, a protective hat to ensure its survival in the cellular crowd. It's spliced, the exons join together, a seamless narrative of the protein it will become. And it's polyadenylated, a poly tail added at the other end, a signal for the next phase of its journey. You notice the ribosomes, tiny factories waiting for their RNA cargo. They're like two subunits, a large and a small, fitting together around the RNA, ready to translate its message into a protein. They read the RNA in groups of 3, each group or codon instructing them to add a specific amino acid to the growing protein chain. It's a precise dance, a rhythm of addition, one amino acid at a time. You notice the regulatory proteins, the activators and repressors, the enhancers and silencers. They're like choreographers influencing the dance of gene expression. Some encourage the transcription, others hinder it. Some work in the background, their influence felt but unseen. They're the silent puppeteers, pulling the strings of gene regulation. This is the intricate dance of gene regulation, a complex system of checks and balances. It's happening right now in the calm landscape of a cell, in every moment, in every generation, in you. And it's been happening for millions of years, a constant, evolving rhythm of life. In the calm, intimate dance of gene expression, you find yourself drifting among the regulatory proteins, their roles as intricate as the cells they inhabit. Silent choreographers, each with its own unique step, influencing the collection of molecular interactions that create life as we know it. You notice the specificity factors, their arms outstretched, reaching for specific DNA sequences, enhancing or inhibiting the transcription of a gene like a ballet dancer pirouetting on command. Maestros of gene regulation, their movements precise, their influence profound. You are now a part of the cellular landscape, the intricate web of life unraveling around you. The regulatory proteins are not solitary figures, but members of an ensemble, each playing its part in the group of gene expression. You notice the repressors, their arms crossed, standing in the way of transcription, hindering the dance of gene expression. Silent sentinels, their influence felt but unseen, their presence a reminder of the constant checks and balances at play in the cellular world. The enhancers and silencers are different, their influence more subtle, their actions more nuanced. Puppeteers pulling the strings from behind the scenes, their actions felt but not seen. Quiet powerhouses of gene regulation, their influence felt in the background, their presence a constant reminder of the delicate balance that is gene expression. You are now a part of the deep past, the timeless pattern of gene regulation playing out around you. These proteins were already at work, their influence shaping the course of evolution. Silent architects of life, their actions written in the very fabric of our being. And they are still at work today, in every cell, in every moment, in you. As the dance of gene regulation unfolds, you find yourself among the transcription factors, their movements precise, their influence profound. The intricate web of life unravels around you, each protein playing its part in the group of gene expression. You notice the repressors standing in the way of transcription, hindering the dance. Silent sentinels, they are felt but unseen, their presence a reminder of the constant checks and balances at play in the cellular world. The enhancers and silencers too are different. Puppeteers pulling strings from behind the scenes, their influence subtle, their actions nuanced. Quiet powerhouses of gene regulation, their presence is a constant reminder of the delicate balance that is gene expression. You drift closer now, your attention drawn to the structure of DNA itself. The double helix twists and turns, an intricate dance of its own, as it winds its way through the nucleus. The packing density of DNA is not just a measure of space, but a regulator of time. Tighter coils slow the dance of transcription, as if the music has slowed, the steps more measured. Looser coils speed the process, like a quickening tempo. You notice the histones, the spools around which DNA winds, their surfaces marked with epigenetic tags, like notes written in the margin of a musical score. These tags, methyl groups, acetyl groups, influence the tempo of the dance, speeding or slowing the process of transcription. As you drift through the cellular landscape, you encounter specificity factors, activators, and repressors, each with its own unique dance. They pirouette and promenade along the DNA strand, their movements guided by the specific sequences they recognize. The repressors, with their arms crossed, move in a slow, deliberate dance, blocking the path of the transcription machinery. The activators, on the other hand, move with a quick, fluid grace, clearing the path for transcription to proceed. The enhancers and silencers too have their own unique dances, their influence felt but not seen, their actions a subtle nudge, a quiet whisper in the grand collection of gene regulation. You are now a part of the deep past, the timeless pattern of gene regulation playing out around you. These regulatory proteins were already at work, their influence shaping the course of evolution. Silent architects of life, their actions written in the very fabric of our being. They are still at work today, in every cell, in every moment, in you. The dance of gene regulation is a story, a tale of life's journey through time, written in the language of biology. And you are a part of that story now, drifting through the cellular landscape, witnessing the dance, the rhythm, the tempo of life itself. As you continue your dance through the cellular landscape, you find yourself in the calm, intricate world of transcription regulation. The DNA strand, a precise highway of information, stretches out before you, its base pairs, the alphabet of life, arranged in a seemingly endless sequence. You notice that the surface of this highway is not smooth, but rather it's pocked with specific sequences like rest stops along a journey. These are the spots where transcription factors, the precise guides of gene expression, take their pause. You see the specificity factors, activators, and repressors, each with its own unique role. They pirouette along the DNA strand, their movements guided by the specific sequences they recognize. The repressors, with their arms crossed, move in a slow, deliberate dance, blocking the path of the transcription machinery. They are the silent sentinels, standing guard over genes, ensuring they remain quiet until the right moment. The activators, on the other hand, move with a quick, fluid grace. They clear the path for transcription to proceed. Their influence felt like a gentle nudge, a quiet whisper in the grand collection of gene regulation. But the dance of gene regulation is not just about the dancers on the stage. In the calm, you notice the enhancers and silencers, their influence felt but not seen. They are the quiet powerhouses, their actions written in the very fabric of our being. They are the silent architects of life, their actions shaping the course of evolution over time and still at work today, in every cell, in every moment, in you. You find yourself drawn to one of these sequences, a specific resting spot for a transcription factor. As you approach, you notice something peculiar. The sequence seems to be covered in a fine white powder. You reach out, your fingers brushing against the powder, and as you do, you realize that it's not just a simple coating, but a collection of methyl groups, acetyl groups, influencing the tempo of the dance, speeding or slowing the process of transcription. This is the quiet, intricate world of epigenetic regulation, where chemical modifications to DNA influence the tempo of life. As you stand there in the calm, you realize that you are not just a witness to this dance, but a part of it. Your every action, every thought, every breath is a result of this timeless pattern of gene regulation playing out around you, within you. And as you drift through the cellular landscape, you are reminded that this dance is not just a process, but a story, a tale of life's journey through time, written in the language of biology. And you are a part of that story now, your dance intertwined with the interaction of life itself. As your fingers brush against the powdery methyl groups, you realize that this coating is a simple marker, but it influences the tempo of life's journey. You are now standing at the intersection of time and biology in the realm of epigenetic regulation. The process of life, a tale of life's journey through time, written in the language of biology. As you take a step further, you notice a structure looming ahead, a scaffold of proteins and nucleic acids. This is the ribosome, the stage where the script of life is translated into the actors that play out our existence. The ribosome is a complex machine, a molecular factory where the instructions carried by mRNA are decoded and turned into proteins. It is composed of two subunits, the large and the small, each a collection of proteins and RNA molecules. They fit together like a jigsaw puzzle, creating a workspace where the magic of life happens. As you approach the ribosome, you notice a cap at the 5' end of the mRNA. It's a marker that plays a crucial role in the translation process. It influences the efficiency and fidelity of translation, ensuring that the script is read accurately and quickly. It's a tiny gatekeeper, a sentry that guards the entrance to the stage, allowing only the right players to take their places. You reach out to touch the cap, but as your fingers get close, you notice something peculiar. The cap seems to be connected to something, a thread that stretches out from it like a leash holding a puppy. This is the polytail, a long string of adenine nucleotides that extends from the 3' end of the mRNA. The polytail influences the stability and translation of the mRNA. The longer the tail, the more time the mRNA has to tell its story before it's degraded and recycled. As you follow the polytail with your eyes, you notice something else. The tail seems to be bound by a protein, a tiny guardian that protects the tail and, by extension, the mRNA itself. It's a shield, a defender that stands between the mRNA and the degrading enzymes, buying it more time to tell its story. But time is a fickle friend, and even with the poly tail and its guardian, the mRNA's story has an expiration date. As the clock ticks down, the poly tail shortens, a little bit at a time. This is a process called detonylation, a slow, steady countdown that signals the end of the mRNA's time on stage. As the tail shortens, the mRNA becomes more susceptible to degradation, its story coming to a close. But even as the mRNA's time on stage draws to a close, its story doesn't end. The proteins it has helped create, the actors it has brought to life, continue to play their roles. They carry out their functions, maintaining the grand journey of life, the grand harmony that plays out around us, within us. And as you stand there, in the calm, you realize that this journey, a tale of life's journey through time, written in the language of biology. And you are a part of that story now, your journey intertwined with the journey of life itself. As the poly tail, now reduced to a mere stub, fades into the distance, your gaze shifts downwards towards the dense spiraling coil that is the chromatin. You are drifting through the twists and turns of the DNA double helix, its sugar-phosphate backbone glistening like a wet dark road under the soft glow of the histone proteins. These proteins, like tiny round boulders, dot the landscape, their tails swaying gently in the calm flow of time. You approach one such histone, its acetylated tails fanning out like the rays of a tiny, pale sun. This is histone H3, and its acetylated tails are like tiny, welcoming arms, beckoning the transcription machinery to come closer. The acetylation has smoothed out the surface of the histone, making it less like the rough rock it once was, and more like a smooth, welcoming pebble on a quiet beach. But this histone is not alone in its transformation. Around it, other histones are undergoing similar changes. Methylation, like a careful, precise tear, is adding tiny, dark flecks to the surface of some histones, while acetylation, like a soothing, warm tide, is smoothing out the surface of others. These modifications, so tiny, so precise, are altering the landscape of the chromatin, changing the way it twists and turns, making some genes more accessible, others less. You are a silent observer in this dance of regulation, this ballet of gene expression. You watch as the chromatin, its structure altered by the epigenetic regulators, unwinds just enough to allow the transcription machinery to pass. The RNA polymerase, like a slow, steady river, flows through the open door, transcribing the information in the DNA into a complementary strand of RNA. But this is not a one-way process. As the RNA polymerase transcribes, the epigenetic regulators continue their work, modifying the histones, altering the structure of the chromatin. It is a constant, dynamic dance, a delicate balance between gene expression and gene silencing. And you, in this quiet, unassuming place, are witness to it all. You notice the chromatin, its structure altered by the epigenetic regulators, unwinding like a slow, gentle wave, revealing the genes hidden within. The transcription machinery, patient and unhurried, approaches, its components shifting and rearranging like a careful puzzle solver. The RNA polymerase, a slow-moving river of protein, flows through. The open door, its path lit by the soft glow of the acetylated histones, like welcoming lanterns along a quiet path. As it transcribes, the information in the DNA unfurls before it a long, winding road of genetic code waiting to be copied into the complementary RNA strand. The RNA. Polymerase moves with a steady, rhythmic pulse, a metronome ticking away the time in the calm dance of gene expression. Around it, the epigenetic regulators continue their work, their modifications to the histones, a collection of tiny, precise changes, altering the landscape of the chromatin. You observe as the methyltransferase, a careful, meticulous artist, adds tiny flecks of methyl groups to the surface of the histones, darkening the landscape like careful brushstrokes on a canvas. Meanwhile, the histone acetyltransferase, like a soothing, warm tide, smooths out the surface of the histones, making the chromatin less dense, more open, more welcoming. To the transcription machinery. As the transcription proceeds, the chromatin, its structure altered by the epigenetic regulators, changes shape and form. Some genes, once hidden, now emerge, their DNA sequences uncoiling, ready to be transcribed. Others, once prominent, now recede, their DNA sequences coiling up, silenced, their information no longer needed. The chromatin is a dynamic, ever-changing landscape, a ballet of gene expression and gene silencing, a delicate balance between the two. You are a silent observer in this dance, a quiet, unassuming presence in the complex machine of the cell. You watch as the transcription machinery moves through the landscape, its path lit by the soft glow of the acetylated histones. The chromatin unwinds and coils, revealing and hiding the genes within. You are a quiet, careful witness to the delicate, intricate dance of gene regulation, a dance that shapes and defines life as we know it. As the methyltransferase's careful brushstrokes fade, the histone acetyltransferase's gentle touch smooths out the surface. The landscape of the chromatin blurs. You find yourself drifting through the shifting terrain, the once distinct edges of the histones losing their definition, merging into a hazy, undulating sea of chromatin. The methyl groups, like tiny flecks of darkness, scatter and disperse, their influence waning. The acetyl groups, once distinct, now blend into the landscape, their impact softening. The chromatin, once a dense, complex canvas, transforms into a vague, shifting expanse, a sea of possibilities, a blur of potential. The DNA sequences, once clearly marked, now merge and blend, their information less apparent, less distinct. Some genes, once prominent, now recede, their DNA sequences coiling up, their information no longer needed, no longer prominent. Others, once hidden, now emerge, their DNA sequences uncoiling, ready to be transcribed, their information now relevant, now necessary. The dance of gene expression and gene silencing continues, but the performers are less distinct, the stage less clear. The transcription machinery, once distinct, now becomes a vague, shadowy presence moving through the landscape, its path lit by a soft, diffused glow. The chromatin, its structure altered by the epigenetic regulators, unwinds and coils, revealing and hiding the genes within, but the process is less clear, less distinct. The dance of gene regulation continues, but the dancers are less clear, their movements less precise. You are a silent, vague observer in this dance, a quiet, unassuming presence in the intricate machine of the cell. You watch, or perhaps sense, the dance of gene regulation, a dance that shapes and defines life, a dance that is less distinct, less clear, but no less important. The dance continues, the music fades, but the rhythm persists, the rhythm of life, the rhythm of the cell, the rhythm of existence, a rhythm that is less distinct, less clear, but no less real. As the dance of gene regulation continues, the once distinct transcription machinery now becomes a vague, shadowy presence moving through the landscape, its path lit by a soft, diffused glow. You are an unassuming presence in this dance, a quiet observer in the grand machine of the cell. You, the chromatin, its structure altered by the epigenetic regulators, unwinds and coils, revealing and hiding the genes within. But the process is less clear, less distinct. The dance of gene regulation continues, but the performers are less clear, their movements less precise. Yet, amidst this blur, specific patterns emerge. You sense a gentle tug, a quiet nudge, as specificity factors drift through the landscape. These factors are not like the activators or repressors, subtle in their approach. They seek out specific DNA sequences, their targets hidden amidst the sprawling expanse of the genome. They find what they're looking for, not through a search, but through a quiet, patient process of elimination. They home in on their targets, not with a burst of energy, but with a slow, steady drift, like a leaf carried by a gentle breeze. Once found, these specificity factors do not assert their presence with the intrusion. No, they simply merge. They blend with the DNA, their information intertwining, their presence becoming one with the gene they found. This integration is a slow, steady process, a gentle evolution over time. The specificity factor does not simply appear and then disappear. No, it becomes a part of the gene, its information now intertwined, its presence now a quiet, persistent influence. This influence is steady, constant. It is a dance that is less distinct, less clear, but no less important. The dance continues. The music fades, but the rhythm persists, a rhythm that is less distinct, less clear, but no less real. In the gentle merging, you find yourself drifting through the quiet embrace of the gene, the specificity factor, now an intimate part of its structure. The edges blur, the details soften as the two become one, their information intertwining like the roots of ancient trees in the earth. This is not the sharp takeover you might expect, but process, a gentle evolution over time. The specificity factor does not simply appear and then become a part of the gene, but its presence is now a quiet, persistent influence, like the steady drip of water from a faucet, constant, unassuming, yet in its persistence. You, the gene, now shaped by the quiet presence of the specificity factor, begins to express itself differently. The machinery of transcription, once complex, now seems more like a well-oiled machine, each part working in harmony with the others, each movement precise and purposeful. The transcription factors, once distinct, now blend with the DNA, their presence becoming one with the gene they found. They are no longer mere visitors, but residents, their influence steady, constant, like the slow beat of a heart in the night. This influence is not, but steady like a candle flame in the dark. It is not a burst of energy, but a quiet, persistent nudge, guiding the cell, shaping its destiny like the gentle current of a river guiding the path of a leaf. This dance of gene regulation, this quiet, persistent influence of the specificity factors, shapes and defines life. It is evolution over time, a gentle, persistent rhythm like the seasons, the tides, the world. The specificity factors, now part of the gene, begin to communicate with each other, their messages not shouted but whispered, like the rustling of leaves in the wind. They do not demand attention but suggest, guide. Their influence, not a command but a quiet, persistent request. The cell, now aware of these quiet whispers, begins to respond. Its machinery of transcription working in harmony with the specificity factors, each part moving in time with the others, like the dancers in a waltz. This dance of gene regulation, this quiet, persistent influence of the specificity factors, is not an intense event, but a gentle, persistent presence. A quiet, constant influence, evolution over time. It is a gentle, persistent nudge, guiding the cell, shaping its destiny, like the gentle guidance of a parent, the gentle influence of a friend, the gentle presence of a lover. It is a dance that is less distinct, less clear, but no less important, no less real. The dance continues, the music fades, but the rhythm persists. Life, the cell, the world, a rhythm that is less distinct, less clear, but no less real. As the specificity factors, now part of the gene, continue their quiet influence, the cell begins to respond. You notice the transcription machinery, once chaotic and uncoordinated, now moving in harmony with these gentle influences. It's as if the cell, once a bustling city of independent workers, has found a gentle guide, guiding each process in time with the others. The specificity factors, like the soft hum of a lullaby, guide the cell's machinery. Their influence, not a command, but a suggestion, a gentle, persistent nudge. This quiet presence, this gentle, persistent influence, is a gradual, persistent shaping, evolution over time. It is not a change, but a gentle, precise nudge, guiding the cell, shaping its destiny. You observe the transcription machinery, once distinct and clear, beginning to lose definition, like a photograph left in the sun. The once sharp lines of the proteins, the once distinct shapes of the RNA, they all start to lose definition, like the setting sun, like the end of a day. The specificity factors, their whispers now quieter, softer, begin to merge with the cell, their influence no longer distinct, no longer clear, but no less real. You notice the cell, once a distinct entity, now beginning to blend with its surroundings. The boundary between the cell and its environment starts to fade, like the line between the sea and the sky at sunset. The cell begins to communicate with its neighbors, their messages not shouted, but whispered, like the rustling of leaves in the wind. This gradual influence, this quiet, constant presence, is a rhythm that persists. Life, the cell, a rhythm that is less distinct, less clear, but no less real. The cell, once a standing figure, now begins to communicate with its neighbors, their messages not shouted but whispered, like the rustling of leaves in the wind. This is a gentle, persistent influence, a quiet, constant presence, evolution over time. The cell, once a distinct, separate entity, is now more a part of its surroundings, a node in a vast, interconnected network. The messages it sends and receives are not loud announcements, but quiet, persistent whispers, like the steady drip of water in a cave. The cell, once isolated, is now part of a greater whole, a collection of cells, each one playing its part, contributing to the overall harmony. The mirage of the DNA, once clear and distinct, now shimmers and wavers like a heat haze. You are floating, weightless, through a sea of chromatin, the once distinct spears of histones. Fading, their outlines becoming less defined. The transcription machinery, once clear and precise, operates with a gentle hum, like the faint echo of a memory. The cell, once a solitary entity, is now part of a vast interconnected web, its boundaries blurring, its messages quiet whispers in the night. The specificity factors, once sharp and distinct, now blend and merge, their specificity less clear but no less real. They drift through the cell, searching for their targets, guided by an ancient innate knowledge. They find their marks, not with announcement, but with a gentle, persistent rhythm, like the steady drip of water in a cave. The repressors, once stern guardians, now stand as softened sentinels, their once rigid boundaries softening, their once unyielding rules becoming gentle suggestions. They no longer stand as barriers, but as guides, their presence more a nudge than a block. The activators, once vibrant energy, now drift through the cell like dreamy dancers, their presence a soft, persistent glow. They no longer burst onto the scene with enthusiasm, but instead they slip into place, their presence more a steady, comforting hum. The enhancers and silencers, once clear sequences, now blend and merge, their roles more complex, more nuanced. They are no longer simple switches, but more like dimmer switches, their influence more a gentle, persistent nudge than a surge. The chromatin, once a tight structure, now unwinds, its surface a rough, textured landscape. The histones, once solid spears, now drift apart, their outlines vague, shifting forms. The DNA, once a clear strand, now becomes a vague, shifting form, like a mirage in the desert. The RNA, once a precise molecule, now has a rough, textured surface. The ribosomes, once clear structures, are now vague, their outlines wavering, their presence more felt than seen. The translation machinery, once clear and precise, operates with a gentle hum, like the faint echo of a memory. The cell, once a distinct entity, is now part of its surroundings, a node in a vast network. It is no longer a single solitary cell, but a point in a complex web of communication and interaction. The messages it sends and receives are not announcements, but quiet whispers, like the steady drip of water in a cave. This is not change, but a gentle, persistent influence, a quiet, constant presence, evolution over time. The cell, once a silent, isolated entity, is now part of a greater whole, a collection of cells, each one playing its part, each one contributing to the overall harmony. The cell, once a distinct entity, is now part of a vast, interconnected system, a system that is not, but gentle, persistent, real. This is the dance of gene regulation, this quiet, persistent influence, not an event, but a gentle, persistent presence, evolution over time. As the last echoes of definition fade, you find yourself adrift in a sea of shifting forms. The once crisp outlines of DNA, RNA, and ribosomes are now vague, their edges blurred like a mirage in the desert. The cell, once a distinct entity, is now more a part of its surroundings, a node in a vast, interconnected network. Its messages are quiet, persistent whispers, like the steady drip of water in a cave. This is not change, but a gentle, persistent influence, a quiet, constant presence, evolution over time. You notice the once rigid structure of DNA unraveling, its once clear, defined strand, now a rough, textured surface. The RNA, once a distinct molecule, is now a vague, shifting form, its once precise structure now a rough, textured surface. The ribosomes, once clear, distinct structures are now vague, their presence more felt than seen. The translation machinery, once clear and precise, is now a distant hum, like the faint echo of a memory. The cell, once a silent, isolated entity, is now part of a greater whole. It is a collection of cells, each one playing its part, each one contributing to the overall harmony. The messages it sends and receives are quiet, persistent whispers, like the steady drip of water in a cave. This is not an event, but a gentle, persistent presence, evolution over time. You drift closer to DNA, now a rough, textured surface. It is no longer a standing figure, but a humming, vibrating entity. It is alive, pulsating with information. It is a vast, interconnected system. It is a web of communication, a network of influence. It is gentle, persistent, real. As you drift closer, you notice the transcription factors. Once distinct entities are now vague, shifting forms. They are quiet, yet insistent. They are not searching, not following, but leaning in, sharing quiet secrets with DNA. They are not predators, not victims, but partners, each contributing to the overall harmony. DNA, once a silent, isolated entity, is now part of a vast, interconnected system. It is a system that is gentle, persistent, real. This is the dance of gene regulation, this quiet, persistent influence, not an event, but a gentle, persistent presence, evolution over time. The transcription machinery hums with life, a gentle, persistent presence. Transcription factors are now vague, shifting forms, murmuring quiet secrets with the DNA. They're not predators, victims, but partners, each contributing to the overall harmony. The chromatin, once solid and compact, is now fluid and dynamic, a bustling city of information. Histone proteins twist and turn, allowing or denying access to the DNA, whispering quiet, insistent voices. The cell, once isolated, is part of a greater whole, a collection of cells playing their part, each contributing to the harmony. The messages it sends and receives are quiet, persistent whispers, not loud announcements. This is evolution over time, a gentle, persistent influence. As the whispers of transcription factors persist, their voices blending into a low hum, you drift through the cell, a vast landscape of information. The DNA, no longer a rigid, unwavering structure, now pulsates with life, its double helix twisting and turning, revealing glimpses of the secrets it holds. The chromatin, once a dense, impenetrable fortress, is now a fluid, dynamic city, its histone proteins acting as gatekeepers, regulating the flow of information. The cell, once isolated, is now part of a greater whole, a collection of cells, each one playing its unique part in the overall harmony. You notice the mRNA, now a persistent, tangible reality. Evolution over time. The quiet stillness of the grove, but a complex network of communication, a network of influence. The ribosomes, once still, now vibrate with life, their precise rhythmic dance a testament to the efficiency of the cell's machinery. The light shifts from a clear, distinct presence to a more muted, diffused glow. The colors, once vibrant and distinct, now blend into one another, creating a sense of harmony and unity. The outlines of the cell, once clear and precise, now blur and merge, creating a sense of fluidity and motion. You hear the quiet hum of the cell's machinery, not a loud announcement, but a quiet, persistent whisper. The messages it sends and receives are not loud announcements, but quiet, persistent whispers. It's like water in a cave, persistent, gentle, real. The epigenome now leans in, sharing a quiet secret with the DNA. The chromatin, now a bustling city of information, a network of influence. The histone proteins, now twist and turn, allowing or denying access to the DNA. They're not silent sentinels, but active participants, their voices quiet yet insistent. The cell, now a tangible, real presence. Evolution over time. The quiet stillness of the grove, but a complex network of communication, a network of influence. The cell, once isolated, is now part of a greater whole, a collection of cells, each one playing its unique part in the overall harmony. In the gentle hum of the cell's machinery, you are drifting through the subtle whispers of information, the epigenome leaning in to share secrets with the DNA. The chromatin, once a vast sea of silence, has become a bustling city of information, a network of influence. The histone proteins, not silent sentinels, twist and turn, allowing or denying access to the DNA, their voices quiet yet insistent. The cell, once isolated, is now part of a greater whole, a collection of cells, each one playing its unique part in the overall harmony. You notice the double helix, once a distant, intangible concept, now leaning in, revealing its secrets. It's not observer, not a subject, but a partner, each contributing to the overall harmony. The DNA, a twisted ladder of information, drifts into view, its rungs whispering tales of inheritance. The bases, once indistinguishable, now reveal their unique identities, a dancing with T, C, waltzing with G, in a slow, steady rhythm of life. The base pairs blend into one another, creating a sense of unity. The outlines of the helix blur and merge, creating a sense of fluidity and motion. The colors blend into one another, creating a sense of harmony. The light softens, casting gentle shadows on the interaction of life. The dance of the base pairs slows, a gentle, persistent rhythm of evolution. The light softens, a quiet, persistent presence. The cell exists, a gentle, persistent presence, evolution over time. The information, a complex network of communication, a network of influence. You hear the quiet hum of the cell's machinery, a quiet whisper. The messages it sends and receives are quiet, persistent whispers. It's like water in a cave, persistent, gentle, real. The epigenome leans in, sharing a quiet secret with the DNA. The chromatin has become a bustling city of information, a network of influence. The histone proteins twist and turn, allowing or denying access to the DNA, their voices quiet yet insistent. The cell is a tangible, real presence. As the light softens, a gentle hum persists, the quiet whisper of the cell's machinery. The dance of the base pair slows, a steady rhythm of evolution over time. You notice the chromatin, once a vast empty sea, bustling with information. The histone proteins, no longer silent sentinels, twist and turn, allowing or denying access to the DNA, their voices quiet yet insistent. The epigenome, once a distant concept, leans in, sharing a quiet secret with the DNA. You are drifting through the open space of the epigenome a living library of information. The histone proteins, like librarians, guide you through the shelves of DNA, their actions dictating what information you can access. You notice the histone proteins, once rigid and unyielding, now flexible and responsive, their actions controlled by chemical signals. You hear the quiet whisper of acetylation, a gentle process that adds a chemical tag to the histone proteins. This tag, like a key, unlocks the DNA, allowing the information within to be accessed. The acetylation, once a slow, steady process, now speeds up, the tags accumulating on the histone proteins like dew on a leaf. The chromatin, once tightly packed, begins to unwind, the DNA unraveling like a ribbon ready to be read. But you also notice the quiet whisper of methylation, a process that adds a different chemical tag to the histone proteins. This tag, like a lock, seals the DNA, preventing the information within from being accessed. The methylation, once a slow, steady process, now slows down, the tags disappearing from the histone proteins like raindrops on a day. The chromatin, once loosely packed, begins to tighten, the DNA coiling like a spring ready to be stored. You notice the histone proteins, once isolated parts, now part of a greater whole. They work together, their actions synchronized like a harmony. The acetylation and methylation, once independent processes, now interplay, their actions complementing each other like a gentle movement. The cell, once an isolated part, now part of a greater whole, the epigenome, a complex network of communication, influence. You hear the quiet hum of the cell's machinery, a persistent, gentle rhythm. The messages it sends and receives are not announcements, but quiet, persistent whispers. It's like water in a cave, persistent, gentle, real. The epigenome, now a tangible presence, shares a quiet secret with the DNA. The chromatin bustling with information influences the cell. The histone proteins, no longer silent, twist and turn, allowing or denying access to the DNA, their voices quiet yet insistent. The cell, now a tangible presence, is part of a greater whole, the epigenome, a living library of information. The interaction of life, now a gentle, persistent rhythm, slows evolution over time. The light, now a soft, persistent presence, softens, a quiet, gentle hum. The cell, now a gentle, persistent presence, is a part of evolution over time. You find yourself in a realm where information is not just a concept but a tangible force. It's a vast, bustling city of knowledge where every street and every house is a piece of the grand puzzle. You're drifting through this city, not as an observer, but as a presence. The streets are the DNA, twisted and turned by the histone proteins, not silent sentinels, but active participants. There are the guardians of the city, allowing or denying access to the houses, their voices quiet yet insistent. The light here is different. It's not the light of day, but a soft, persistent glow. It's like the light in a deep-sea trench, soft and constant, guiding the way. It's the light of understanding, illuminating the path through the city's labyrinthine streets. You notice the messages. They're not loud announcements, but quiet, persistent whispers. They're the city's heartbeat, a gentle, rhythmic pulse. It's like the sound of water in a cave, persistent, gentle, real. It echoes through the streets, carrying information from one house to another, from one gene to another. You hear a quiet hum. It's the city's machinery, the transcription machinery. It's not, but a gentle, persistent rhythm. It's the sound of life, the sound of evolution over time. It's the sound of the city growing, changing, adapting. As you drift through the city, you notice the specificity factors. They're like the city's librarians, each one specializing in a specific house, a specific gene. They're not just passive guardians, but active participants in the city's life. They're the ones who enhance or inhibit the transcription, the ones who decide which houses get their light and which ones don't. You see them, the specificity factors, leaning in towards the DNA, sharing a quiet secret. It's like they're whispering, this gene should be expressed now, or this gene should be silenced. Their voices are quiet, but their influence is profound. They shape the city, they shape life. The light softens, the messages persist, the hum continues. The city is a living library of information, a complex network of communication. It's a web you're part of, a web that influences you, a web that you influence. The interaction of life is now slow, a gentle, persistent rhythm of evolution over time. The city, once the event, is now a gentle, persistent presence. The light, once, is now soft. The messages, once loud, are now quiet, persistent whispers. The hum, once, is now a gentle, persistent rhythm. The city, once distant, is now a tangible, real presence. You are part of this presence, part of this web, part of this dance. You're still drifting, the city's hum a persistent, quiet rhythm. You notice the light now, soft, a gentle persistence. It's like the sun has grown. Tired, its rays no longer sharp, but a warm, comforting embrace. The messages it carries are no longer loud exclamations, but quiet whispers, persistent and patient. They're like the city's librarians too, each one specializing in a specific house, a specific gene. They're not just passive guardians, but active participants in the city's life. They're the ones who enhance or inhibit the transcription, the ones who decide which houses get their light and which ones don't. But there's more to this city, more to these librarians. They don't just guard the information, they transform it. The light, it changes. It's not just about the light that's produced, but the light that's absorbed, the light that's reflected. It's about information, life, and in this dance, there are certain steps that are particularly important. They're called post-transcriptional regulation, and they're the focus of our attention now. As you drift, you notice the specificity factors. They're still there, leaning in towards the DNA, sharing their quiet secrets. But now you see them do something else. They're not just whispering, this gene should be expressed now, or this gene should be silenced. They're also modifying the message, changing it in subtle but profound ways. They're capping it, splicing it, adding a polytail. It's like they're taking the raw manuscript of life and turning it into a book that's ready to be read. Capping, that's the first step. Imagine the specificity factors as editors sitting at their desks, a stack of manuscripts in front of them. They take the first one, the raw transcript of a gene, and they add a cap to it. It's like the editor's signature, a mark that says, "This is ready to be read." But it's not just any cap. It's a 7-methylguanosine cap, a special mark that tells the cell's machinery where to start reading. It's a signal, a command, a gentle nudge towards translation. And then there's splicing. The editors, they're still at their desks, but now they're working with a different set of tools. They're using their scissors, cutting out the parts of the manuscript that don't need to be in the final book. These are the introns, the parts that don't carry any information, the parts that need to be removed. And then they're using their glue, sticking the remaining parts, the exons, together. It's like they're creating a new story, a new version of the gene, one that's ready to be expressed. It's a process that's happening all the time, all around you, in every cell of your body. It's a process that's as constant and as persistent as the city's hum, as the light's gentle glow. And finally, there's polyadenylation. The editors, they're not done yet. they take the edited manuscript, the one that's been capped and spliced, and they add a poly tail to it. It's like they're adding a final chapter to the book, a chapter that's not about the story itself, but about how the story should be told. This tail, it's a signal too, a command that tells the cell's machinery how long the book should be read. It's a signal that's as important as the cap, as important as the splicing. It's a signal that's as persistent and as gentle as the city's hum, as the light's soft glow. And so, life continues. The city, it's not just a gentle, persistent presence anymore. It's a constant, persistent hum, a gentle glow. The messages, they're not just quiet, persistent whispers anymore. They're quiet, persistent, and patient. The hum, it's not just a gentle, persistent rhythm anymore. It's a constant, gentle hum, a persistent presence. And you're part of this presence, part of this web, part of this dance. Your city light messages, You're part of life, information, post-transcriptional regulation. In the gentle glow, you notice the editors, they're not done yet. They take the manuscript, the one that's been capped and spliced, and they add a poly tail to it. It's like they're adding a final chapter to the book, a chapter that's not about the story itself, about how the story should be told. This tail, it's a signal too, a command that tells the cell's machinery how long the book should be read. It's a signal that's as persistent and as gentle as the city's hum, as the light's soft glow. You're drifting through the cell. Now, the city of life, where messages are whispered, not shouted. The polytail, it's a quiet presence, a persistent signal that's as important as the cap, as important as the splicing. It's a signal that guides the cell's machinery, telling it when to start reading the book and when to stop. The tail, it's not just a simple string of adenine nucleotides. It's a complex structure, a dance of its own. It's got a protective cap at the end, a cap that shields it from the cell's hungry enzymes. And it's got a length that varies, that tells the machinery how long the story should be told. The longer the tail, the longer the story. The shorter the tail, the quicker the story ends. You notice the editors now, they're not just adding the tail, they're also trimming it. They're removing adenine nucleotides from the end of the tail, a process called dedenylation. It's like they're editing the final chapter, making sure it's just the right length. And as the tail shortens, the cell's machinery gets the message. The story's almost over. It's time to stop reading. The cell, it's not event anymore. It's a gentle, persistent presence, evolution over time. The messages, they're not just whispers. They're quiet, persistent signals. The polytail, it's not just. It's a gentle, persistent signal. And you're part of this presence, part of this web, part of this dance. You're part of the city, the light, the messages, the hum. You're part of life, information, post-transcriptional regulation. The cell's machinery, it's not just an event either. It's a gentle, persistent presence too. It's reading the book, the one with the polytail, the one that's telling the story of life. It's reading it slowly, carefully, making sure it doesn't miss a word. And as it reads, it's translating the story into a protein that's essential for life. The protein, it's not just an event. It's a gentle, persistent presence, A presence that's as constant and as persistent as the city's hum, as the light's soft glow. It's a presence that's essential for life, a presence that's the result of a long, complex process. A process that starts with a gene that's read, edited, and expressed. As the polytail shortens, the cell's machinery begins to lose interest in the message. It's like a storyteller who's been reciting the same tale for too long, their voice fading, their audience drifting away. The tale, once vibrant and pulsating, now whispers. It's not, but a quiet, steady decay. The cell, it's not an event, but a presence, evolution over time. The messages, they're not just and loud, they're quiet and persistent. The polytail, it's a signal, and you're part of this presence, part of this web, part of this process. You're part of the city, the messages, the hum. You're part of life, information, post-transcriptional regulation. The cell's machinery, it's not just an event, it's a presence too. It's reading the book, the one with the polytail, slowly and carefully. As it reads, it's translating the story into a protein essential for life. The protein, it's a presence, a constant and persistent hum. It's essential for life, the result of a long, complex process. A process that starts with a gene, read, edited, and expressed. This process is persistent and gentle, happening all around us in every living thing, from the smallest microbe to the largest tree in the ocean, city, and natural world. It's the process of life, information, and post-transcriptional regulation. In the soft glow of the city at night, you find yourself drifting through the quiet hum of the environment. The gentle rustle of the leaves, the soft patter of the rain, the whisper of the wind, they're all persistent rhythm. It's happening all around us all the time. The process of life, information, regulation. You notice the polytail, a long string of adenine nucleotides extending from the 3 end of the mRNA. It's a fragile thing, like the first few notes of a harmony, barely perceptible but essential. It's a protective measure, a safeguard against the inevitable decay that awaits all things. The poly-binding protein, a tiny, dedicated servant, clings to this tail, protecting it, buying it more time to tell its story. But even this loyal servant can't prevent the inevitable. Dedenylation, the quiet, steady shortening of the polytail, is a process as constant and as persistent as the city's hum. It's a fading away, a slow, steady loss of meaning, a story winding down to its inevitable end. But even as the polytail shortens, gene expression continues. The story of life goes on, even as the words fade away. The regulatory factors, the specificity factors, the activators and repressors, they're all still there, still working, still making sure that the right genes are turned on, the right proteins are produced. It's a complex balance, a harmony that plays on without pause, without rest. And as you drift through this silent, unseen world, you realize that you're part of it. Your cells, your body, it's all vast, interconnected web of life. Every living thing, from the smallest microbe to the largest tree, is part of this process. It's happening all around us, all the time, in the soft glow of the street lights, in the gentle rustle of the leaves, in the calm hum of the environment. It's life, information, regulation, And as you drift through this silent, unseen world, you realize that you're not just an observer. You're a part of this harmony that plays on without end. In the soft, fading glow of the streetlights, you continue to drift through this silent, unseen world. The polytail, once so vibrant and full of life, is now a mere shadow of its former self. Dedenylation has taken its toll, the quiet, steady shortening of the tail, a process as constant and as persistent as the city's hum. Yet, even as the tail fades, life goes on. The transcription factors, those dedicated servants, are still at work, clinging to the remnants of the tail, protecting it, buying it more time to tell its story. But even their efforts are not infinite. The polytail continues to shorten, the words of the mRNA story growing fainter with each passing moment. You notice the polybinding protein, that tiny, loyal servant clinging to the tail, its job growing increasingly difficult. It's a fading away, a slow, steady loss of meaning, a story winding down to its inevitable end. Yet, even as the polytail shortens, gene expression continues. The story of life goes on. The harmony plays on without pause, without rest. The activators and repressors, those conductors of the group, are still there, still working, still making sure that the right genes are turned on, the right proteins are produced. It's a complex dance, a delicate balance, a harmony that plays on without end. But you also notice something else, something new. There's a quiet, persistent hum, a gentle rustling in the air. It's a message, a signal, a silent call to action. It's a whisper, a fading away, a story waiting to be told. It's the specificity factors, those messengers of the genome carrying their silent, unseen messages from one gene to another. They're the postmen of the cell, dedicated to their task, focused on their mission. They're the ones who make sure that the right messages get to the right place at the right time. They're the ones who ensure that the collection of life continues to play even as the polytail fades away. You drift closer, drawn to this quiet, persistent hum. You notice that the specificity factors are not alone. They're part of a vast, interconnected web, a network of messages and responses, a complex dance of signals and reactions. They're part of a system, a machine, a vast, unseen world of life. They're part of you, part of me, part of every living thing. They're the quiet, persistent hum of life, the soft, fading glow of the streetlights, the gentle rustle of the leaves, the quiet, steady shortening of the polytail. They're the process, the story, the harmony that plays on without end. And as you drift through this silent, unseen world, you realize that you're not just an observer. You're a participant. You're part of the process, the story, the harmony. Your life, information, post-transcriptional regulation, and as the polytail fades away, you know that the story is far from over. As the polytail fades, you notice a subtle shift in the air. It's not the rustling of specificity factors anymore, but a different kind of quiet. It's the silence between notes, the pause before the next phrase, the stillness that follows a gentle breeze. You're drifting now, not through a collection of life, but through the spaces between them. The silence is not empty. It's full of potential, of waiting, of the next message sent. You find yourself in a still expanse. It's the space between cells, the gap between genes on a chromosome. It's the distance between you and the next thought, the pause before the next word. It's a quiet, still place, but it's not silent. as a hum, a gentle, persistent hum. It's not the hum of life, but the hum of something else. It's the hum of potential, of waiting, of the next message sent. You're not alone in this stillness. There are others here, like you, waiting, listening. They're not specificity factors, not repressors, not activators. They're not even transcription factors. They're something else, something different. They're the ones who listen for the silence, who wait for the pause, who send messages when the time is right. They're the ones who understand the quiet, who speak the language of stillness. One of them turns to you. It's not a cell, not a gene, not even a protein. It's a shape, a form, a presence. It's a messenger, a postman, a standing figure. It leans in, not to whisper, but to listen. It listens to the silence, to the stillness, to the pause before the next thought. It listens for the message that's sent, for the story told. What do you hear? It asks, not with words, but with a gesture, a tilt of the head, a raise of an eyebrow. It's a question, but not one that expects an answer. It's a question that invites you to listen, to wait, to understand the quiet. It's a question that asks you to join in the stillness, to become a part of the pause, to help send the next message. You listen. You wait. You understand the quiet. You become a part of the stillness, a participant in the pause. And as the silence deepens, as the stillness becomes more profound, you realize that the story is far from over. The message is sent. The next note is about to be played. The collection of life is about to resume. And you, in your stillness, in your silence, are a part of it all. You are the pause before the next note, the silence between the words, the stillness that follows the breeze. You are the process, the story, the harmony that plays on without end. And as the silence deepens, you know that the story is far from over. Sleep well and good night. In the soft hush of fading light, the final note of journey sounds. As we come to end of exploration, landscape before us stretches wide, open space of quiet contemplation. The day's warmth leaving behind gentle caress of cooling air, last whisper of breeze through leaves. The world around us, once bustling with life, now settles into deep, profound stillness. The polytail begins slow, steady shortening. It's a quiet process, unassuming yet crucial, like steady drip of leaky faucet. In silent night, the poly-binding protein, a tiny, tireless sentinel, clings to tail, protecting it, buying more time to tell its story. But even most devoted guardian must eventually relent. The dedenylation complex approaches, its presence heralded by subtle shift in air, softening of silence. It's a question, not of if, but of when. The question, of course, is not loud, demanding one. It's a quiet, persistent query, like soft rustle of leaves or gentle hum of life that persists even in stillest of moments. It's a question that doesn't expect answer, but rather invites you to join in quiet, to become part of process. As silence deepens, stillness becomes more profound. We realize that story is far from over. The message is sent, next note is about to be played, collection of life is about to resume. And we, in our stillness, in our silence, are part of it. All. We are pause before next note, silence between words, stillness that follows breeze. We are quiet, persistent hum of life, soft, fading glow of streetlights, gentle rustle of leaves, quiet, steady shortening of polytail. Now, as final note of journey fades, you can let those ideas soften, let them drift away like last wisps of dream upon waking. For now, rest is enough. Sleep well.